Alrighty, hello biology students. So I am out today, so what I'm going to have you do is start preparing for a lab that we're going to do next time I'm in class next week. So hopefully from the sub you have received this piece of paper. Let me make this a little bit bigger. So you should receive this lab, this Inquiry with Isopods Lab Part 1 for biology. So this is what you're going to be working on today and preparing this so that you can do the lab next class. So before you get started in this video, I want you to go ahead and pause it in a moment. But at the very first question, it says use the space below to record any knowledge you have about isopods or pill bugs. So a pill bug, you might have called it a roly-poly, or like one of those little critters that rolls into a ball when you touch them. That's called an isopod. So I want you to write everything you know about an isopod right now as you pause the video. Try to get down at least 3 to 5 things that you already know. It could be their color, their shape, how they behave, where they live, anything that you know about isopods. Okay, after that we are going to go through a little bit of introductory information about isopods. So go ahead and just follow along with your packet, and as you follow along, just kind of fill in notes as you go. So I'm going to do a little bit of teaching about isopods so that you have a little bit of background information before you start planning a lab. So, some things about isopods. An isopod has 2 large antennae on its head, so that's how they kind of feel where they're going, how they can see. Isopods also have a greater brown exoskeleton. An exoskeleton is basically a hard outer shell that protects them. As hopefully all of you have seen one, you kind of know what that looks like. If you haven't seen one yet, hopefully some of your classmates have already started to bring isopods in so that you can kind of start observing them. Go ahead and grab one from the middle desk and start checking them out so you can make some other observations on them. And they also have 7 pairs of jointed legs, so 14 legs in total. So they have a decent amount of legs. Pill bugs are classified in the subphylum Crustacea. So in biology, every living critter is divided into these different phylum of like classes and orders and species and stuff like that. And so isopods are in the subphylum Crustacea. And you know of some other common animals that are also known as crustaceans, which include lobsters and crabs and even shrimp. So notice some similarities between these little pill bugs or isopods and a lobster, crab, and shrimp. Notice they have those hard exoskeletons. They have more than one set of legs here if you're a crab, but they have some antennae there as well. So they are all closely related since they're all in the same subphylum. So even though these are all water-dwelling creatures, the isopod still has a lot of similar characteristics. So ethology. Any idea what the word ethology means? Notice it has that ending ology, and we know that ology means the study of. So ethology is the study of animal behavior. So we're going to be studying the behavior of isopods and trying to figure out what they like more or less than other things in this lab. So, for example, when pill bugs are scared, they curl into a ball. That's something an ethologist would notice. They're studying that when pill bugs get nervous, they curl into a ball. So, what you're going to be doing now is hopefully someone has brought in a pill bug. If not, you'll have to look one up online, or ideally someone has brought one in and was on top of things. I want you to try to answer these questions as you start observing a pill bug. How do they sense their environment? Any ideas on how they do that? Think about how you can tell the difference between males and females. How many eyes do they have? Try to notice these things while you're observing a pill bug. So I'm going to have you grab a pill bug out of the center table and then place them in front of you at the end of this video. Please make sure that they don't escape. So what you're going to be using is you have these little choice chambers. So you're going to have something that looks like this, and this is called a choice chamber, and they sit like this. So you're going to put the pill bugs into either of these chambers, and then the pill bugs will be able to be like stuck in here. They do come with covers, but don't completely cover them like this. That won't let the pill bugs out, which is good because they can crawl out of those edges, so make sure you're watching them closely. So if they get out, just pick them up, put them back in. But make sure you do provide them some air, so maybe like vent it just a little bit like this so that they can still have some fresh air. Okay, but go ahead and grab a pill bug and place it in the choice chamber and start observing the pill bug. With this, you're going to make some detailed observations in the form of a drawing. Okay, biological drawings are really important and are used in a lot of things. So think back to like way long ago when cameras weren't a thing. Everything was based on drawings, so we're going to be drawing our pill bug here today. So be super detailed, include its exoskeleton, include color if you want. You need to label at least 5 different parts of the isopod. So make sure you label them, describe them, show detail in your picture. Okay. When you're handling the pill bugs, so make sure you're using stirring rods or little pokers to move the pill bugs around. Don't try to grab them too fiercely because you can crush them because your fingers are much bigger than their little bodies. Make sure you keep the choice chamber in the center of the table. Don't set it on the side of a table in case a partner or a friend bumps it off as they're walking past. We don't want them to tumble to the floor and spill all over. And make sure you're using soft voices so you don't frighten the pill bugs and you're being respectful to Mrs. Johnson. So that is all I have for you as far as background information for the lab. So now I'm going to go into what is expected of you and what you're going to be doing for the rest of this class period. Okay, so on this second page here, you have a chart, and it has you put a question, hypothesis, dependent variable, control, and constants. So what your job is going to be to do is decide what question you want to test so that you can learn something about pill bugs. Okay, so it can be any question. Maybe it could be something about the type of environment a pill bug prefers, or the type of food they would prefer, or the type of temperature they would like to prefer. You can do any of these questions or anything else that you can think of. Okay, so your job is to create a testable question. After you form a testable question, you must form a hypothesis, and remember that needs to be an if, then, because format. You need to identify your independent variables, your dependent variables, and more importantly, your control. So what are you going to be holding steady during this experiment? And sorry, those are your constants, but your control is what is normal in this experiment. So what are you going to be comparing your results to? The major part of today is actually after you decide what you want to test, you need to form a procedure on how you're going to test this lab. This needs to be a super detailed procedure. So if I come in after being absent and I try to perform your procedure, I can perform it exactly how you performed it. It needs to be able to be repeated. It needs to be detailed. You need to include what materials you're using and everything. There is a materials list listed on Canvas of just some things I'll be providing to you. In addition to that, if you want anything else, go ahead and ask me and I'll provide you with most of the things you need, or you can bring some stuff in from home as well if you want to test something a little bit obscure. Okay, so after you form your investigation plan or your procedure, make sure you have a data table ready to collect your information. So make sure with your group you develop what type of data you're going to be collecting. Think about, do you want to collect qualitative data or quantitative data? Do you want to have something with numbers or something with viewing? How are you going to collect that data? How often are you going to collect that data? And how are you going to display it so it's easily readable or legible? Okay, so these are all things you need to think about so that you are prepared next class to test your experiment as soon as you get to class. Because if you don't finish the experiment in class next time, you're going to have to stay after school and finish it. Okay, so if you don't finish planning your experiment in class all the way, that is homework. However, if you do finish, there is another POGL for you to work on that you started last class, and you can go ahead and finish that up. But be ready to test your lab next class. If you have any questions, reach out to Mrs. Johnson. If you have any additional questions that are like urgent that you need to have asked before starting the lab, make sure you find Mrs. Clausen. She's the other teacher that teaches biology in room 106, and she can help you with whatever you need. You can email me as well, but I can't guarantee I'll respond right away since I'll be on vacation. But I look forward to seeing you guys next week and seeing what kind of experiments you come up with. Bye.